Welcome to Showbiz Insights. Let's take a look on the keynotes of vaccination, which is explained in a simplified way. Vaccination means the act of introducing a vaccine into the body to produce protection from a specific disease. First, about the principles of vaccination. The principle of vaccination is to induce protection against a pathogen by mimicking its natural interaction with the human immune system. The vaccine reduces the risk of complications and mortality following subsequent exposure to an infectious agent. Secondly, antibody-mediated protection. Antibodies produced as a result of immunization are effective mostly against extracellular organisms and their products. Example, toxins. Passively administered antibodies have the same effect as induced antibodies. Thirdly, about cell-mediated immunity. Vaccination induced cell-mediated immunity by T cells, macrophages is important particularly in preventing intracellular bacterial and viral infections and fungal infections. Immunization. Immunization is the process by which a person becomes protected against a disease through vaccination. There are mainly four types of immunization. The first one is passive immunization. Passive immunization is the administration of preformed antibodies either intravenously or intramuscularly. It is used to provide rapid protection in certain infections such as diphtheria or tetanus or in the event of accidental exposure to certain pathogens such as hepatitis B. It is also used to provide protection in immune-compromised individuals. Second type of immunization is active immunization. Active immunization is the administration of vaccines containing microbial products with or without adjuvants in order to obtain long-term immunological protection against the offending microbe. The third one among the immunization is systemic immunization. At present, the normal route of vaccination in most instances is either intramuscularly or subcutaneously. The fourth way of immunization is mucosal immunization. Oral immunization is the method of choice for polio and Salmonella typhi vaccines. However, there is an increasing awareness that this route of immunization may be the best for most immunization since nearly all infectious agents gain entrance through the mucosal surfaces. The third factor related to vaccination is antigen. What is antigen? An antigen is any substance that causes your immune system to produce antibodies against it. This means your immune system does not recognize the substance and is trying to fight it off. An antigen may be a substance from the environment such as chemicals, bacteria, viruses or pollen. The first one to mention about antigen is antigen preparations. Protection against pathogenic microorganisms requires the generation of effective immune mechanisms. Thus, vaccines must be capable of targeting the immune system appropriately, that is, cellular and humoral, by plasma cells mechanisms. Continuation from antigen preparations. Most vaccines consist of either attenuated organisms, killed organisms, inactivated toxins, subcellular fragments, genes for antigens in viral vectors and DNA itself. The third one worth mentioning about antigens is adjuvants. Adjuvants are non-living vaccines, especially those consisting of small molecules requiring the inclusion of agents to enhance their effectiveness. These adjuvants include microbial, synthetic and endogenous preparations having adjuvant activity. But at present, only aluminum or calcium salts are generally used in humans. Continuation of adjuvants. Adjuvants should enable antigens to be slowly released, preserve antigen integrity, target antigen presenting cells, and induce cytotoxic lymphocytes. The third one in antigen category is DNA vaccines. The use of DNA encoding antigens as vaccines is distinct from bacteria or bacterial proteins has shown potential. Intramuscular injection of circular DNA results in DNA uptake by muscle cells, expression of the encoded protein, and induction of both humoral and cell-mediated immunity. Recombinant vaccines are the fourth one among antigens. Using molecular genetics, selective recombinant proteins of defined epitopes can be prepared that protect the host. This approach overcomes the problem of disease complications which might occur with modified live vaccines. The fifth one worth mentioning among antigens is cytokines. Cytokines can be added at the time of immunization to skew the immune response to a Th1 or Th2 type depending on which is associated with protection. The cytokines can be added either as purified protein made from recombinant technology or they can be cloned into the vectors to be delivered at the time of vaccination. Cytokines that might be useful are interleukin 12 or interferon gamma which favor a Th1 response or interleukin 4 and interleukin 10 which favor a Th2 response. What is Th1 and Th2 cytokines? The Th1 and Th2 cells play an important role in immunity. The Th1 cells stimulate a cellular immune response whereas Th2 stimulates a humoral immune response. What are cytokines? Any of a number of substances such as interferon, interleukin, glycoproteins, and growth factors which are secreted by certain cells of the immune system and have an effect on other cells. What are interferons? A protein released by animal cells, usually in response to the entry of a virus, which has the property of inhibiting virus replication. What are polypeptides? A linear organic polymer consisting of a large number of amino acid residues bonded together in a chain, forming part of or the wall of a protein molecule. What are interleukins? Interleukin 12 is a heterodimeric cytokine. Interleukin 10 is a cytokine with potent anti-inflammatory properties that plays a central role in limiting host immune response to pathogens, thereby preventing damage to the host. Interleukin 4 is a cytokine that functions as a potent regulator of immunity. What are interferons? Interferon gamma or type 2 interferon is a cytokine that is critical for innate and adaptive immunity against viral, some bacterial and protozoan infections. The fourth type of vaccine is the vaccines against pathogens and tumors. What are tumor cell vaccines? A vaccine composed of tumor cells isolated from the tumor of one patient, killed and processed and administered to another patient in order to stimulate cytotoxic immune responses to a similar tumor cell type. Bacterial vaccines. Bacterial vaccines have been developed for many different types of bacteria: Escherichia, Haemophilus, Pneumococcus, Vibrio, Helicobacter, and Lyme's disease spirochete to name a few. Perhaps more familiar is diphtheria, pertussis, and tetanus vaccine that many young children receive to protect them from often fatal childhood diseases. Viral vaccines. Vaccines have been developed to viruses that infect the respiratory tract, the gastrointestinal tract, the skin, and some that infect the reproductive tract. As with bacteria, viral vaccines are either modified, live, killed, or subunit. Vaccines to other infectious agents. Protozoan parasites such as those that cause malaria like Plasmodium, African sleeping sickness like Trypanosoma, and schistosomiasis are major diseases mostly of the third world. The ability to vaccinate people and animals against protozoan diseases will allow people to live in areas that are endemic, where the organism is always present for the disease. Tumor vaccines. Vaccination strategies against cancer are currently being investigated. Vaccines containing tumor antigens such as those associated with prostate cancer as well as those associated with breast, colon and ovarian cancers such as HER2 often hope for the future. That's the end of keynotes on vaccination. Stay immune.